All right, AP Biology folks, let's do it. This is the last set of notes for Unit 1. It's all about lipids. And here's those notes. Okay, lipids are super great. They are used mostly for energy storage. They are also in cell membranes. I've also added here steroids, if I know how to spell it correctly, which are like messengers. So their structure, the big thing about them is that they are non-polar. They are hydrophobic. They do not interact. They don't interact with water. Well, that's not strictly true. As a chemist, I can, chemistry teacher, I cannot straight up tell you that because really it's more like the water interacts with itself more than it wants to interact with the fats. So there's a whole video. I'll put it on the list. It's great. It explains it. So aside from that, the other thing that you need to know about lipids is that saturation is a really important factor for them, or property. Saturated refers to how many hydrogens are bonded to them. So lipids are hydrocarbons. So they're made of almost exclusively carbon and hydrogen. Hydrocarbons are also what make up like fuels, like fossil fuels are made of hydrocarbons. The big difference with fatty acids or lipids is that they have some sort of end cap on them that kind of alters their structure. I'm going to show you some pictures out of the textbook that I think are helpful for that. So saturated is one, there's kind of four words you need to know. There's saturated, unsaturated, monounsaturated, and polyunsaturated. Saturated means that the carbon chain is completely full of hydrogens. So I've got a nice example right here. There it is. You can see the carbon, every carbon is bonded to 4 other molecules, except for this one, which there should be a double bond in there, but whatever, it's fine. So maybe this is not the best picture, whatever, it's okay. So yeah, you can see that each carbon is bonded either to 2 or 3 hydrogens. So they're all completely full. Unsaturated means that there is a place where one of those hydrogens has been removed and is replaced with a double bond. So actually in this case, sorry, it's 2 hydrogens. So here is a place, this is a monounsaturated fat, where you can see here, there's the 2 hydrogens over here that should have been attached are removed, and instead this carbon and carbon are double bonded. This creates a different structure. So instead of being a relatively straight chain, it now has a bend or a kink is what we usually call those. A polyunsaturated fat has multiple places of those kinks or bends. And that changes a lot of their properties, especially in terms of like how much they can stack. So something like lipids that are kind of straight, don't have those kinks that are fully saturated, can stack on top of each other very easily. As soon as they start having bends, it's hard, they can stack, but it's less likely to happen. So kind of neat. Let me show you all. Should I go into that? Yeah, we'll do this first. We'll talk about phospholipids. So phospholipids are a specific type of lipid that have a phosphate group on them, hence the phospholipid. So this is their structure. They have 3 fatty acid chains. I'll show you more pictures of fatty acids, but that's usually how biological lipids are constructed. They have these chains of lipids that are attached to something that is polar, some sort of structure that kind of holds them together. Otherwise, they wouldn't really be able to interact with a lot of biological molecules because, you know, we are basically big bags of water, like big tubes full of little pockets of water. So yeah, that's important. Yeah, so a phospholipid essentially has this head that is a phosphate base. There's the phosphate right there. You'll remember phosphates from nucleic acids. Yeah, the phosphate sugar backbone thing. So this part is polar. You can tell that because it's got this positively charged nitrogen, there's a negatively charged oxygen, there's a negatively charged oxygens here. There's just lots of nitrogens and oxygens and phosphates, which are super polar, hydrophilic, all that deliciousness. The tails, though, are hydrophobic because they are made of carbons and hydrogens. So the way that this ends up working is the general trend is that hydrophobic things attract each other and hydrophilic things attract water or each other. So if you throw a whole bunch of these into water, they will naturally form one of these two kinds of shapes, either a little circle where all the hydrophobic tails are in the middle, hiding away from the water, and all the hydrophilic heads are on the outside, hanging out with the water, or a phospholipid bilayer, which is these two layers. This is, of course, what cell membranes are. So I usually say that I don't really think of lipids as being monomers and polymers like a lot of the other macromolecules. I guess they kind of are. Like if you think of these chains, these fatty acid chains as the monomers, and to polymerize, they kind of connect there, but not really. Also, they're not, you don't use dehydration synthesis to create those extra bonds. These are actually more ester bonds. So they don't really form monomers and polymers. This is sort of like a monomer and polymer, but not really, because none of these are bound together. The fact that they're not bound together is really important because it's extremely fluid. All of these parts can just freely flow because they're not really bound together. It's just like attracted to each other. So that's kind of important and pretty dope. Phospholipid bilayers. Okay, let's look at the book real quick and just see if there's anything interesting. I think I saw a few interesting diagrams. In terms of what you need to know, I think that's good enough. But just for some extra information, let's go into this. So glycerol is this, it's kind of like glycogen or glycerol. I don't know if it's a sugar. I think it's a sugar. That looks like a sugar to me, actually. But it can bond to these. Oh my gosh, it is a dehydration reaction. I was wrong. It's like a polymer. Oh my gosh, everything is a dehydration synthesis. This is amazing. Okay, so cool. Okay, so here's a fatty acid. You can tell that because it's got all these hydrogens and carbons. It's got one little end that will attach to this glycerol. This is a triglyceride. You may have heard of those before because it's a glycerol with these 3 fatty acid chains. Pretty cool. They're all different lengths, so that makes a really big difference too. There's different kinds of oils and different kinds of fats, and their properties are all determined a lot by the chains and if they're hydrated, saturated or unsaturated. So for example, these are all fully saturated, and I know that because I don't see any double bonds at all. Yeah, let me just see if there's a double bond on the other page. Yeah, there is. There's a couple. Okay, so in terms of saturated and unsaturated, there's a very nice picture over here that helps explain it. There, right there is that double bond that causes that weird little kink. So there you go. Definitely makes some effects. There's a lab related to this, so we'll go into that more later. Here's some more information about phospholipids. They also talk about trans fats here, which I didn't talk about explicitly, but we could always have a discussion. Let me know. Here's another phospholipid diagram. Gives you the same basic idea. Hydrophilic head, hydrophobic tails. It's a good way to draw it. Pretty simple. And there's your phospholipid bilayer. Oh, we should talk steroids and cholesterol. So lipids also create these other types of molecules that have, like this is kind of the lipidy, the super lipid part, and then here's just like more hydrocarbon rings. So that is cholesterol, which is a type of steroid. Steroids are usually messengers. They're more for cellular communication. That's their main purpose. And that's the other thing that lipids are used for. You'll notice with lipids that you don't really need, you definitely don't need nitrogen. And you can have steroids without phosphorus also. You do need phosphorus for the membranes because of that phosphate head. That's that yellow molecule, or that little, yeah, the yellow dot right there. Okay, so that's pretty cool. Awesome. So let me show you some videos. Actually, let me show you some on Teams, some resources that might be useful for you about lipids. I don't, honestly, this is not an area of chemistry that I've done a huge amount of studying. Okay, so, yep, there's lipids notes right here. So if you want to learn more about lipids, they're cool. Just remember they're hydrophobic. That's the main thing. If you can remember they're hydrophobic, and saturation's nice. I don't know how often that comes up, but hydrophobic is the main thing. Let's look at assignments. There is a membrane structure POGL, so that could be very cool. That'll be about lipids. And yeah, not too much else in here. There's a lab though, so let's check that out. Okay, this is cooking fats. This is great. I do this lab with my AP chemistry students too. So here's what you're going to do. I want you to compare the molecular structure of different fats and to see how those structures affect, like dictate their function and properties. So what I want you to do is grab in your kitchen as many cooking oils as you want. I think at least 3 is good. You've got things that I had in my cabinet. I have canola oil and coconut oil and sesame oil, and I don't have vegetable oil actually. I have olive oil, butter, lard, shortening, margarine, schmaltz, which is, it's a Yiddish word for chicken fat. Tallow is beef fat. Lard is specifically for pig fat. Little lesson for you there. Any of that stuff, if you've got earth balance, that's like margarine. Anything that you could cook with that's an oil or a fat. Just get a little sample of each. You don't need a lot. And what I want you to do is put it in either little cups, or you could take little bits of aluminum foil and make like little pockets for it. You could use ice cube trays. If you've got those little takeout plastic cups, those are great. Anything that you have lying around that you think would be okay. Just take a small amount and just put it in those containers, and just take a look at them. See what they're like. What do they look like? Possibly what they taste like. Any other observations about them. And then what you're going to do is put them in the freezer overnight, or the refrigerator, but the freezer is better if you've got space. Pop them in the freezer overnight, and the next day, look at them and see what has happened. And you can make notes about that. So make sure that you, also definitely clean up after this. It's going to be a lot of mess. Use that soap. So the questions that I have for you are to research what types of fatty acids are in them. It shouldn't be too hard if you put in like for example, olive oil molecular structure. You should come up with some results. The main thing you want to look for is saturation. How saturated is it? And if you can get a picture, that's better. And I want you to draw your own model or select one of those pictures to show. And also use those pictures and also written explanations for how the structure affects the properties. It might have something to do with it getting cold. That's a hint. Something will happen when it gets cold, like freezing kind of thing. Anyway, also, the last question is that generally speaking, health professionals say it's probably a good idea to limit your saturated fats. And why, based on what you have seen and observed in this lab, why do you think that that might be true? So what would be like some negatives to eating lots of saturated fats? Could be interesting. I have one video here that you could watch that might be useful, but let me see what else I have on YouTube for you. I might have something else. Let's find out. I don't even have that one on this list. I'm gonna have to add it. Oh, there's a video about lipids from Mr. Anderson. That's great. No, it doesn't look like I have it. I'll have to add that video. Here is also the video that contains some more information about lipids from AP Biology from the College Board from last year. So those are a whole bunch of resources for lipids. Hopefully that helps elucidate what you need to know. Again, the notes are just abbreviating everything, so you don't have to know all of this. There's just key things to remember. All right, that is the last of my videos. Yes. Oh, so good. I'm going to make some more for labs, or we'll talk more tomorrow and in the next couple days about all of this stuff. You have assignments to work on. If you've gotten to this far in the videos, you probably have your test coming up fairly soon. And we'll talk more about that. As always, please let me know if you need anything. Help each other out. Be excellent to each other. And I will catch you on the flip side.